Today I want to tell you a story about a classical biological control story. This is an importation biological control having to do with the control of weeds. This is a different kind of pest. This is a plant pest rather than an insect pest. And the control happens by the introduction of herbivores, which suppress the population of the pest. But let me start here with this beautiful plant that's worldwide that you can see behind me, the prickly pear cactus. These are plants, cacti in the genus Opuntia. It's a New World genus, so-called New World genus, occurs in the Americas from South America, Argentina, all the way up into, actually you can find them in Wisconsin as well. But there are several areas, centers of endemism that center on Mexico and South America. Because of the various attributes of Opuntia, this plant has been moved around all over the world. One of the features that it has is that this plant is colonized by a little insect, a scale insect, that is referred to as the cochineal bug, the cochineal scale. And the cochineal scale, you can see kind of the waxy secretions of the scale here that it uses to protect itself. The scale can actually be removed from the pads and used to make a dye, a red-colored dye. It's here you can see some of the infestations on the prickly pear pads. These are called cladodes. And again, you can see kind of the waxy secretions, and the scales occur underneath those secretions. This is a feature that, an attribute that has been used for hundreds, if not millennia, of years by local populations such as the Inca and groups in Mexico to make these wonderful dyes and garments that they would use to dye wool. The British, in their colonial expansion, recognized this, and they made their army jackets in this red color. That's why we call them, the British, the Redcoats. And during their period of expansion and imperialism, as they moved into Asia, into India, and into Australia, they moved the prickly pear with them so as to establish local industry and supply of cochineal dye. It's thought that the first introduction into Australia was in the late 1700s, 1787 here, and it wasn't long before prickly pear really made itself through the entire country here. After a relatively benign period of maybe 80 to 80 years, the prickly pear really started to spread rapidly into areas that it wasn't introduced into originally. Not only was it used for cochineal dye, but the farmers and ranchers there also used it for fencing. They would kind of plant it in little lines where they then could put their animals in there. It has spines, so animals don't like to eat it, and they could kind of be kept in there. So this is a convenient way to pen your animals up without having to put up things like barbed wire, which was not really invented until after the early 1800s. But of course, plants don't sit still, and this is what happened after a relatively short amount of time. If you count 100 years, a short amount of time, it really expanded into the interior, making millions and millions of acres completely unusable for livestock. Here's some of these would grow quite tall, and it was established as a pest by the local government. This is an article from 1922. There was legislation that was passed to try to control prickly pear, including, first an acknowledgement that this was a problem by the late 1800s. There was the Prickly Pear Destruction Act in 1886 that made landowners responsible for destroying the plant. In 1901, the Queensland government offered a reward for people who attempted to destroy the prickly pear. And here you can see a picture of one attempt to actually control it using chemical methods. This was so-called Robert's Improved Pear Poison, where this was a boiling hot mixture of sulfuric acid and arsenic that kind of wafted out of these carts and was supposed and kind of poisoned and was caustic and damaging to the plants. And as you can imagine, this was only good very near where these boiling pots actually were, and not and couldn't be used to control vast swaths of prickly pear in the outback. There were other methods of control, including kind of mechanical clearance, hand harvesting, and the recognition that various animals were responsible for spreading the seeds of Opuntia, including emus, which you can see here, crows, magpies, and so on. And there was a bounty put on these animals that the government would pay for. So hundreds of thousands of emus were slaughtered to try to slow down the spread of this cactus pest. By 1925, the total infested area was over 24 million hectares. This is well over 50 million acres. As you can imagine, the idea of biological control was starting to take hold, and folks recognize that there's the possibility that maybe there's some herbivores that could be used to control the expansion of this particular pest. The Queensland Prickly Pear Traveling Commission was formed to try to find potential biological control agents. This was not really successful. In 1920, a new board, the Commonwealth Prickly Pear Board, was established, which then retraced the steps of the commission and collected over 50 different species of insects to bring back to Australia to explore them for potential use against prickly pear, against Opuntia. Eventually there was a breakthrough where a specific insect, Cactoblastis cactorum, it's got a wonderful name, which had previously failed its tests for introduction, actually was able to establish a breeding population of adult moths in Australia, and was shown to be host specific. That is, it only fed on Opuntia. It bred prolifically, and it actually laid its eggs in a series of little egg sticks that could be used to transport and release them in various parts of where prickly pear was established. This is what the moth looks like. Here it's doing a headstand. You can see it laying its eggs as a little stick here. Each one of these little rings, it's hard to see the segments, each one of these little rings is actually a single egg. The sticks might have 50 to 100 eggs perhaps. Here's a photo that I took out in the field just recently. They look like the spines on a cactus, which is kind of interesting. And the larvae are these very brightly colored orange and black pyralids. The Cactoblastis moth was unbelievably successful at suppressing the population of Opuntia. Here you can see kind of a before and after picture. This is the same signpost here, C37, the same couple of trees, and the moth absolutely laid waste to these populations. And even today, it's considered maintained well under control by Cactoblastis. It's not completely gone, Opuntia that is, but it's maintained at significantly lower levels. Let's fast forward 30 years since the introduction of Cactoblastis into Australia to this idyllic little island of Nevis and St. Kitts in the Caribbean. The farmers there were having trouble with Opuntia in their pastures. They had goats, primarily sheep in some areas. It's a small industry, and they looked to Cactoblastis for control of Opuntia. They released the moth there in 1957. Here's where Nevis and St. Kitts are in the Antilles here, and not too far from Puerto Rico and obviously the U.S. Mainland. Now, the issue with the introduction into Nevis and St. Kitts is that this is an area where Opuntia is actually native to. This is a movement of the moth from southern, the moth that is native to to South America, to the southern parts of the continent, to an area north of the equator that has other Opuntia species that actually are susceptible to Cactoblastis, where Cactoblastis is not normally found. This is also an area with frequent hurricanes, and it was found that the moths from Nevis and St. Kitts here have a high likelihood of being transported to the neighboring islands here through hurricanes that blow the adult moths from one island to the next. There's also, to a lesser extent, the movement of Opuntia for ornamental and horticultural reasons among the islands. It was not shown that the moth itself is very capable of just hopscotching from island to island just by flight alone. So over time, this moth has been able to actually spread into away from this original area of release into other areas. When you look at the potential suitable areas where this moth can be found based on climate envelopes, this is where Cactoblastis is originally native to, and everything that is kind of dark here are places that have comparable climates that would allow the moth to actually survive and reproduce. And one of these areas here is the Caribbean and the southern US and parts of Mexico. It's also quite suitable here in South Africa and Australia, obviously where it was released into parts of Kenya and so on. And these are actually where introductions of the moth have been made. There was also an introduction into Hawaii. This was an intentional introduction where there are no native Opuntia here. So certainly moving the moth from here into any of these areas would mean that there's a high likelihood that it would spread because of the suitable climates. And in fact, this is what was observed. The moth appeared here in Florida Keys in 1989, and slowly over time it spread northwards and along the Gulf Coast as well. It had reached Dauphin Island in Alabama by 2004. So this is, approaching, 20 years ago, not quite. And just last year, it was actually found having crossed the Mississippi, which is one of the kind of barriers that folks were hoping that it wouldn't cross. And now it's kind of hopscotched past Houston into Brazoria County. Now, this area here of the southwestern U.S. is an area with a high level of endemism, and in particular in northern Mexico. And it was also found in the Yucatan Peninsula in 2019, where a very intense and concerted effort to control it was embarked upon. And from what I can tell, it was successfully eradicated from the Yucatan through a process of kind of scouting and hand collecting and destroying of the moth wherever it was actually found. This is again where Opuntia species are actually found in the U.S. Lots of endemism here in the desert Southwest, and in particular in Texas as well, not to mention Mexico. Where I am right now actually is here in coastal Georgia on Sapelo Island, one of the places where in the year 2000 the moth was found. What I want to do next is actually take you out with me in the field to actually see these infestations and see what Opuntia looks like in these areas.